Don't freeze.
I never freeze.
The revolution will not be televised.
There are four main macromolecules in the body: carbohydrates, proteins, lipids, and nucleic acids. These first three will be discussed in other videos, but this video will focus on nucleic acids, specifically DNA and RNA. As you may have heard, DNA is the chemical blueprint for our body. Using just 4 different subunits in adenine, guanine, cytosine, and thymine, it codes for the over 100,000 different proteins in the body. To understand how this is done, let's take a look at a different coding system, the English alphabet. In the way that an old, fragile book might be housed in a library, DNA is kept locked up in the nuclei of cells. To prevent damage to such an old book, people read copies of it. Analogously, slightly different copies of DNA called RNA are formed and exported from the nucleus for reading. The book only contains the 26 letters of the English alphabet, and DNA only has the 4 subunits, or nitrogenous bases, described earlier. If we put letters together, we can form a word, but the word itself doesn't have meaning. It takes someone who speaks English to read the word and interpret it. In RNA, a set of 3 adjacent bases is called a codon. The codon itself doesn't have a meaning until a tRNA molecule reads it and matches it up with a protein subunit, or amino acid. Once we have words, we can put them together to make a functional sentence. In an analogous manner, adjacent codons make up a gene, and the corresponding amino acids are put together to make a protein. This protein then folds up and performs actions based on its reactivity. DNA and RNA even have start and stop codons on either side of a gene, the same way we use capital letters and punctuation to start and stop a sentence. But imagine if someone came in and changed, added, or erased a letter. Depending on the change, the sentence may still make perfect sense, some sense, or it might be completely nonsensical. In the same way, a mutation in DNA can result in a protein that is either still functional, partially functional, or completely dysfunctional. If the protein is fairly unimportant, a mutation may not cause any disease. If the protein is vital, a mutation can cause a severe disease.
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Say to you, it's in your genes. And we're talking about your pants. Ha! No, it meant you were born that way. It's in your genes. Now, genes tell your cells what to do. Like, you're a stomach cell. You're a brain cell. You're gonna have brown eyes and a big nose. Now, genes are made of DNA, deoxyribonucleic acid.
What?
Deoxyribonucleic acid.
What?
Deoxyribonucleic acid.
Okay.
And DNA molecules are very long molecules that have chemicals on them in a sequence, in a special order. Now, parts of DNA molecules are what we call genes. Now, DNA molecules are packed in your cells in something called chromosomes. Now, humans have 23 pairs of chromosomes. You get 23 from your mom and 23 from your dad. That's,
46?
46.
Uh-huh.
Now, all plant and animal cells have chromosomes. They have different numbers depending on which plant or animal you're dealing with. DNA is pretty cool when you think about it. In fact, thinking's in your genes, too.
You have about 20,000 genes in your DNA. They encode the molecules that make up your body, from the keratin in your toenails, to the collagen at the tip of your nose, to the dopamine surging around inside your brain. Other species have genes of their own. A spider has genes for spider silk. An oak tree has genes for chlorophyll, which turns sunlight into wood. So where did all those genes come from? It depends on the gene. Scientists suspect that life started on Earth about 4 billion years ago. The early life forms were primitive microbes, with a basic set of genes for the basic tasks required to stay alive. They passed down those basic genes to their offspring through billions of generations. Some of them still do the same jobs in our cells today, like copying DNA. But none of those microbes had genes for spider silk or dopamine. There are a lot more genes on Earth today than there were back then. It turns out that a lot of those extra genes were born from mistakes. Each time a cell divides, it makes new copies of its DNA. Sometimes it accidentally copies the same stretch of DNA twice. In the process, it may make an extra copy of one of its genes. At first, the extra gene works the same as the original one. But over the generations, it may pick up new mutations. Those mutations may change how the new gene works, and that new gene may duplicate again. A surprising number of our mutated genes emerged more recently, many in just the past few million years. The youngest evolved after our own species broke off from our cousins, the apes. While it may take over 1 million years for a single gene to give rise to a whole family of genes, scientists are finding that once the new genes evolve, they can quickly take on essential functions. For example, we have hundreds of genes for the proteins in our noses that grab odor molecules. The mutations let them grab different molecules, giving us the power to perceive trillions of different smells. Sometimes mutations have a bigger effect on new copies of genes. They may cause a gene to make its protein in a different organ, or at a different time of life. Or the protein may start doing a different job altogether. In snakes, for example, there's a gene that makes a protein for killing bacteria. Long ago, the gene duplicated, and the new copy mutated. That mutation changed the signal in the gene about where it should make its protein. Instead of becoming active in the snake's pancreas, it started making this bacteria-killing protein in the snake's mouth. So when the snake bit its prey, This enzyme got into the animal's wound. And when this protein proved to have a harmful effect and helped the snake catch more prey, it became favored. So now what was a gene in the pancreas makes a venom in the mouth that kills the snake's prey. And there are even more incredible ways to make a new gene. The DNA of animals and plants and other species contain huge stretches without any protein-coding genes. As far as scientists can tell, it's mostly random sequences of genetic gibberish that serve no function. These stretches of DNA sometimes mutate, just like genes do. Sometimes those mutations turn the DNA into a place where a cell can start reading it. Suddenly, the cell is making a new protein. At first, the protein may be useless or even harmful, but more mutations can change the shape of the protein. The protein may start doing something useful, something that makes an organism healthier, stronger, better able to reproduce. Scientists have found these new genes at work in many parts of animal bodies. So our 20,000 genes have many origins, from the origin of life to new genes still coming into existence from scratch. As long as life is here on Earth, it will be making new genes.